Hello students, good morning. I am your educator Dolcy and today we are going to do the most important topic from your chapter human reproduction, that is the structure of the human spermatozoon or the human sperm. So first of all you should be familiar with what is spermatozoon. Spermatozoon or the sperms are motile, means they are capable of moving from one place to another. Microscopic means they can be observed only under the microscope. Structures or the cells which remain alive for about 24 to 48 hours after being reached in the female genital tract at the time of copulation. So, I am repeating, the sperms are motile microscopic structures which remain alive in the female genitalia for about 24 to 48 hours after the act of copulation. Means during the act of copulation, the male adds its sperms into the genitalia of the female, that is through the vagina into the uterine canal, and in that canal it remains alive for 24 to 48 hours. So the most important from this is the structure of the spermatozoon. As you know, during spermiogenesis, the spermatids, which are spherical structures, get converted into elongated spindle-shaped structure that are said to be the spermatozoa. So this one is the complex diagram of the spermatozoon. The spermatozoon primarily is divided into four main parts. First is said to be the head, second is said to be the neck, third one is middle piece, and final one is the tail. Usually head comprises of two parts. First is said to be the acrosome, second is said to be the nucleus, which contains the genetic material which is transferred into the nuclei of the female ova where it undergoes fertilization. Second part is the neck. It is the area which carries two centrioles. First one is the proximal and second one is the distal centrioles, which are required for the division of the zygote after fertilization. Next one is the middle piece. Middle piece is the area where mitochondrial spiral is present. Mitochondrial spiral is the spiral or spring shaped structure found in the middle piece formed of the mitochondria. As you know, mitochondria are the powerhouse of the cell. So the function of the mitochondrial spiral here is to provide energy so that the sperm can move. Final one is the tail. Tail is comprised of fibrous sheath in it, and this type of tail is responsible for the movement of the sperm in the female genitalia. And at the end, the piece is exposed which lacks the fibrous sheath, that is said to be the end piece. And most important of these is the acrosome. Acrosome is formed by condensation of the Golgi complex. Means as you know, during the transformation of spermatid into spermatozoon, certain kind of activities are involved which gather up the Golgi complex or the Golgi bodies at the tip of the nuclei. So the acrosome comprises of a number of hydrolytic enzyme, and it is the kind of structure which is responsible for the penetration of the nucleus into the female ova. So acrosome is very necessary for the degradation of the membrane to ensure the fertilization. So first structure is the spermatozoon is head. Head is the elongated end of the sperm which contains the nucleus and the acrosome. As the acrosome contains number of enzymes which are used to contact and penetrate the egg at the time of the fertilization. Next is the neck. Neck is very short, means it is just small condensation or compression of present at the base of the head and at the initiation of the middle piece. It contains two centriole. First is proximal centriole and second one said to be the distal centriole. Proximal centriole is present towards the nucleus side and it plays a very important role in the first cleavage of the zygote, means from one cell to the two cell stage. Second one is said to be the distal centriole, which give rise to the axial filament of the sperm and which is important for the flip flash movement of the sperms. Next is the middle piece. It is the broader part, more broader than the neck, and it comprises of the mitochondria that are coiled around the axial filament. So it is known as the mitochondrial membrane. The mitochondria, they provide energy to the sperm so that they can move from one place to the another. At the end of the middle piece, the ring centriole is present. This ring centriole is very important and it is known as annulus, but its function is not known till date. As you have observed that half of the nucleus, then the neck and then the middle piece, they are surrounded by a sheath or a covering that is said to be the Manschette's membrane. Next one is the tail. Tail is several times longer than the head. In its most part, it is said to be the main piece, and at the end, when the main piece is naked, it is said to be the end piece. So, if the axial sheath or a fibrous sheath is present, then it is said to be the main piece. If that sheath is absent or the filament is naked, then it is said to be the end piece. So, the sperms can fluid or swim in the genital tract of female only with the help of this tail. So next and most important structure from this is the structure of the ovum or the oocyte. So as it is shown, it is comprises of a mature ovum. Ovum is the female genital tract, genital cell or the gonad or gamete, which is spherical in shape. And I hope you are familiar with this, that human or mammalian, specifically human ova, it is alacithal type. It is of alacithal type, means it has lack of yolk, means the yolk is absent from the human ova. So that is said to be the alacithal type of egg. The cytoplasm of the egg is said to be the ooplasm. There is large sized nucleus present in the almost center of the ova. Then it is surrounded by the plasma membrane, and small cortical granules are present inside the cytoplasm. Then out of this outer side of the plasma membrane is the perivitelline space that is present outside the plasma membrane. Perivitelline space is quite thick in size. Between this, the polar bodies are present, which are formed during the meiosis 1 and meiosis 2 type of divisions. Outer to the perivitelline space, there is a thin layer that is said to be the zona pellucida, which is necessary for the fertilization. And ultimately, and the outer one is said to be the corona radiata, which surrounds the zona pellucida. So, the zona pellucida is the non-cellular type of membrane, probably which is secreted by the follicular cells. Outer to this zona pellucida is a thick layer that is said to be the zona corona radiata. As you know, there is no yolk, so there is no division of the vegetal and animal pole in the human ova. But at the side at which the polar bodies are present are said to be the animal pole, and opposite to this is said to be the vegetal pole. Human ova loads its ability to fertilize. As you know, the fertilization of the ova, it occurs at the 14th day of the menstrual cycle, and that release of ova from the female genitalia is said to be the ovulation. And the viability period of the human ova is 24 hours after the ovulation. And the released ova from that stage of ovulation is said to be the secondary oocyte. So the male sperm is quite longer or quite thread in shape, while the ovum is spherical in shape. Sperms are formed from 4 sperms are formed from spermatogonia, but only one ova is formed from the one oogonia. There is differentiation of head, neck and tail in the sperm, but there is no such differentiation in structure in the ovum. Cytoplasm is very small in the sperm, but ooplasm is very large in quantity in females. Mitochondrial spiral is present in the middle piece of the male, but mitochondria are scattered in the cell. And keep this in mind, the mitochondria of the male are not involved at the time of fertilization. So the mitochondria always exhibit maternal inheritance, means mitochondria are always inherited from human mother to the baby. So it is said to be the uniparental type of inheritance in which the female inherit the mitochondria to its baby. That's why mitochondrial activity of the offspring is always related with the mother, not with the father. Sperms are motile and ovum are immotile. Next, some similarities are also present. So, as you know, spermatogenesis, it comprises of three phases. Similarly, oogenesis also comprises of three phases. The growth is always initiated from the oogonial type of cells. Both the cells have food reserve, and all the cells undergo two type of division. First is meiosis 1 and meiosis 2, and ultimately result in formation of the gametes. So today we have done the structure, differences and similarities between spermatogenesis and oogenesis. Tomorrow we will see the kind of dissimilarities found in spermatogenesis and oogenesis, and how the cells gain the specific type of shape of sperm after undergoing meiosis or the spermatogenesis. Thank you.